And I think because we just have this 500-year-old dogma that these are ancient, simple critters, that every time we or others discover some miraculous property of bacteria, our immediate knee-jerk reaction is, bacteria can't do that. And then we're always wrong. And so that's actually very joyful to always be wrong about that.
Let me begin with your journey as a scientist. I mean, your brush with science began when you were just 13 years old, I'm told. In fact, you wanted to be a veterinarian and joined University of California at Davis to do major in veterinary science. But then your career took a slightly different turn and you ended up doing biochemistry. Would love you to tell us about your remarkable journey from an aspiring veterinarian to a path-breaking biochemist now, whose work has totally revolutionized the way we perceive and think about bacteria and the microbial world in general, with potential applications in the future for the humanity. Yeah.
Yes. So my path, I would say, was unpredictable. And as you described, growing up, I had never met a scientist. I wouldn't have known what a scientist did. And I loved the natural world. I loved animals. I loved puzzles. I loved games. But I didn't know how to put that into something that would be a career. And because I loved animals, my parents thought, oh, she must want to be a vet. She must want to be a vet. And so I was sort of pushed in that direction. And of course, it was a long time ago. Girls didn't become doctors. My family would have never known what a scientist did. And so I went to college after volunteering for these jobs, in the summers and working with animals, I went to college to be a vet. And I was terrible at memorizing, like the bones, the blood system. I was just terrible at it. And I actually didn't enjoy the classes. I didn't enjoy anatomy. Couldn't take the blood, basically. But what was super lucky for me was that as part of that curriculum, you had to take chemistry and biochemistry and genetics and molecular biology. And I fell in love with those classes because you didn't have to memorize. You could figure out the answer by, it was sort of puzzle doing. And so I switched my major to biochemistry. And so I volunteered in this professor's lab. He happened to put me on this bacterial project. And I just thought it was the most amazing way to spend my time. It was sort of doing puzzles and solving mysteries and trying to make discoveries. And I had no idea how thrilling it could feel to make a discovery. But even those little mini discoveries that were part of a much bigger project, I got such a rush from that. And I just loved working at the bench. And so I didn't want to get a job. And so I thought I'll go to graduate school because that's a way that I can keep doing this. And so I went to graduate school and I worked on bacteria. And then by then I was completely committed to a life in research. If I could somehow figure out how to make that happen. I thought, why would I ever do anything but this? Because it was fun, it was challenging, and I felt like maybe learning about life would make a contribution, that it could be helpful for people. So the serendipitous part was that at the end of my graduate career, again, I didn't have a plan, and I hope that students who are listening to this don't think this was some strategic straight path. I didn't have a plan. And I got to go to my very first science meeting. And I was sitting in the audience listening to all these, it happened to be in Baltimore, which is where I was in graduate school. And I was listening to all these talks, and mostly I couldn't understand what anyone was talking about. These scientists would get up and they would give these talks that after the second slide, I was lost. Or they would give these talks that I would think, why would you ever work on that? That doesn't even sound important. You can't even make that sound important for this 20-minute talk. And so I remember sitting there thinking, maybe I don't even like science. Maybe this isn't for me. And then this man got up, his name is going to be Mike Silverman. So he got up and he told this story. Gave this seminar about this beautiful bioluminescent bacteria that only made light when the bacteria were in a group. And he was trying to figure out how could a bacterium know when it's alone and when it's in a group and do something as a collective. And I remember sitting there being mesmerized by this talk because I'd been working on bacteria since I was 19. By now I was like 26 or 27. And I'm thinking, I never heard of that. Like these bacteria are doing this group behavior. And then he also said, he's a geneticist. And he said, look, we can figure this out by making mutants and just turning the lights off in the room and seeing which bacteria are dark and which ones are bright, and we'll be able to find the components by doing this simple experiment. And I remember sitting there thinking, surely I am smart enough to turn the lights off in the room and look on the Petri plates and see which ones are bright and which ones are not glowing. And so I literally ran up to the podium and I begged him. I was like hanging off the podium and I begged him to let me be his postdoc. And eventually he said yes. But when I think back to that moment, I can't believe that I had the courage to get up and ask for what I wanted, right? I had never even like raised my hand and like asked a question at a seminar. I was so intimidated. I had such imposter syndrome. And somehow this young woman, I don't really remember her, but it was me. Somehow she got up and asked him to give her a job.
But interestingly, you've also been an aerobics instructor for a longer period than you've been a biochemist.
Correct.
So, I mean, how do you sort of, look back and say that you continue to do that? You continue to be an aerobics teacher at the same time. You're also doing a biochemistry. You are such an energetic person. You exude energy all the time, including when you're giving a lecture and so on. I mean, does that energy come from doing aerobics all the time?
So doing the aerobics, that's you have to exercise. And the way that started, it was actually when I was in graduate school. I had moved to Baltimore, to Johns Hopkins, and I didn't have any money. It was the first time I ever lived in a big city. And in Baltimore, it's really hot in the summer and it snows in the winter and it's a very busy city. So you couldn't just go outside and jog or play tennis. And I didn't have enough money to join the gym. And so I got this job teaching aerobics because then I would exercise and I didn't have to pay for it, right? And it forced me, and I mean forced me, to get up every morning and exercise, even if I was tired or had experiments to do. If you're the teacher, you must show up. And so I realized that having that sort of framework was really good for me. So I did that for 35 years. And of course I did love it. And I met all kinds of friends and it's very social. Obviously I'm a very social person. I work on social behaviors. I work in a lab with a group of people. I like doing social things. But partly it was because it makes me show up. Even if it's like freezing or snowing or I'm tired, I would show up. Yeah, but it was really fun. And
I believe
you have to exercise.
Also sing and dance and so on.
Yeah. So
you also dabble in theater.
Yeah, dabbled in theater. I'm actually so I don't teach aerobics anymore because the place went bankrupt during COVID, when everything shut down. And so I retired, in air quotes, at that point. But my husband and I are avid ballroom dancers. So we ballroom dance 3 or 4 nights a week. Right. And on weekends. Yeah. And so that is my real passion. Yeah.
Okay. In fact, while giving a talk, I must say that you're actually a real performer there on the stage. I mean, that's really something must be coming out of your dancing and singing skills, I'm sure.
Yeah, I hope so. But also, actually, to that point, I have been let's call it criticized a little bit, about that, like these sort of everyman talks, these blue collar talks that are very simple. I tell a lot of jokes, I'm high energy. I mean, the high energy, that's just me, right? I can't, if you made me like keep my hands by my side, I could not give that talk. But the part about like it being simple and being excited, like, why wouldn't I be? Like I think there used to be this the culture in science that you had to be very serious and, the smartest person in the room, but it was kind of boring, right? And so I think that like my job, people are devoting an hour to sit there and listen to me talk about my gang science. Like we're having fun in the lab. We're making discoveries. I think they're important discoveries. I think we are changing the way people think about how bacteria live and the profound importance of them in nature and medicine, but we're also having fun. I work on glow-in-the-dark bacteria. I don't think I have to be that serious when I give these talks. I mean, that's what my life is about, right? And so I hope that people, like I think that I want people to enjoy listening to science like a mystery story, the same way when you listen to a television program, you want to know, what we're going to get to. But in our case, it's the discovery.
One learned in school and college that these are basically tiny, hard to see with naked eyes, single-celled organisms, what you call prokaryotes, that are reclusive and standalone kind, which just keep dividing and proliferating, and occasionally cause deadly diseases in you. But we also know that they kind of, are beneficial to us. They help us make yogurt, cheese, wine, and so on. And of late, we've also learned that these things help us digest our food, make our vitamins, and also clean up our environment, like oil slicks and so on. But what you have done is you wondered, where do these rod-shaped, wiggly little things get their power from? And you set about finding out about them. And your work over the last 3 decades and more has led us to a big paradigm shift in the way we perceive and view these creatures. You find them that they are doing their own kind of aerobics, if you may. Okay, right? They use molecular mechanisms to talk to each other by exchanging chemicals, what you call them as autoinducers. Through this mode of intercellular communication, they build colonies, cooperative, to do things collectively, and show extraordinary social traits, much like higher organisms like us, which you call eukaryotes, right? Can you take us through your work, all of which began, as you just said some time back, with this innocuous glowing bioluminescent bacteria called Vibrio fischeri?
So what my gang in my lab always wonder is how bacteria get their power. I mean, they're puny, they're tiny, they're the first, most ancient organisms on Earth. You can't see them with your eyes, and yet they do all these miraculous things, harmful and beneficial. And so we've always wondered, how can they manage that? And so what we showed, starting with this bioluminescent bacterium, which started this field, is bacteria have to be able to understand when they're alone and when they're in groups, and they need to behave differently under those two situations. And so what they do is that they make and release small molecules that we call autoinducers. And so as the bacteria grow in number, as they divide in number, the more cells there are, the more of these molecules there will be in the surroundings. And when the molecules hit a particular amount, the bacteria detect them, and they infer from that detection event that they must have neighbors around. And so in unison, all of the bacteria change their behavior together, and they begin to carry out tasks that it takes lots of bacteria acting together to make the task successful, because it couldn't be successful if a single bacterium did it alone, because they're too tiny to make a difference. And we call that process quorum sensing. So they sense when they have the quorum, which is the right number, right, to do something that you can't do with fewer. And so that started in these bioluminescent bacterium because they turned on bioluminescence. They turned on this beautiful blue light that the scientists, we could just see it with our eyes. So the bacteria would grow, they would make no light, then they would all turn on light together. And then we found these autoinducers, we showed that it was chemical, and fast forward over these 30 years, right now we get that that is the norm in the bacterial world. There are tens of thousands of cases of quorum-sensing bacteria, including globally important pathogens, bacteria that are important in the microbiome to keep us healthy. You already brought it up, bacteria that do bioremediation, that clean up oil slicks, bacteria in agriculture, that they have to be able to synchronize tasks to make the tasks successful. And so now we get that this is the first social behavior on Earth because bacteria are the most ancient. And then maybe what I'll also tell you is that we also now know there's multiple molecules involved. If you want to think of these molecules as chemical words, they have a molecule that says, you are my identical twin. They have a molecule that says you're related to me, but you're not identical, like you're my cousin. And then they have molecules that say you're the enemy. And they measure these different blends of molecules, and they decide what to do moment to moment to moment, depending on how many bacteria are around them and also who is around them. Is it friend or is it foe? And then they change their behavior when they're surrounded by their kin and their friends. They do all kinds of nice behaviors. And when they're surrounded by the enemy, they try to hurt each other and they try to take things away and they compete. And it should sound very familiar to things we know about multicellular organisms, right? And of course, we think that these rules and principles evolved in bacteria billions of years ago.
But in your initial days, when you actually did these revelatory findings, even the scientific community found it hard to accept.
Yeah.
They thought it was a one-off thing, and you said that you had to wait for 13 years to get an NIH grant. Mm-hmm. How was that initial period of uncertainty in your life?
Especially for the young people, again, like my non-strategic path, right? It's not strategic to decide to work on a bioluminescent bacteria that's never even heard of fish, right? That nobody's ever heard of, right? But of course, those decisions were impulsive, but they were good. They were good in retrospect. It worked out. Obviously, I'm sitting with you. It worked out. But you're right. The beginning of this field was tough because scientists and human beings have known about bacteria for 500 years. And it was always thought for 470 of those years, it was always thought that bacteria were so primitive, right? They're single cells, they have one piece of DNA, you can't see them. And if you watch them, they just divide in half and one becomes two, becomes four, and so on. And so they were always thought to not have the wherewithal to do sort of fancy behaviors. So bacteria gave us the parts list. DNA, RNA, proteins, we know those from bacteria. And then the fancy stuff, behaviors, architecture of the body, all of that was in eukaryotes. And so here I come along with Mike Silverman, right? This is now 30 few years ago. I'm this young woman, and we have this idea that oh no, maybe these bacteria can act in groups. It was ridiculous, right? Because it's hard to change dogma. And so we only had one example, this bioluminescent bacteria that had this trait, light production, that wasn't important for medicine, for agriculture, for industry. And so even though I think that scientists believed our data and that this glow-in-the-dark bacteria could do this group behavior, they thought it was a one-off, that wasn't going to be medically relevant or wasn't going to be general in the bacterial world. And so it was hard, right? Because I was sort of considered, on the fringe of science. I don't blame the funding agencies, for not funding me because there was no underpinning to what I was doing at the beginning. And so it was really lucky for me is that somehow Princeton University hired me. And Princeton's a really special school. We don't have a medical school. And so everyone in my department works on fundamental problems about life. So in Drosophila, in worms, in yeast, in bacteria. And so somehow they took a shine to this project, right? Now, they had a lot of microbiologists there that thought she might be onto something. And then they backed me during the hard times, right? They gave me resources so I could have equipment in my lab. They funneled graduate students, brave graduate students that would come work for me as some untested assistant professor with this fringy project. And then together, we just kept making discoveries and publishing them, which is the currency that we work by. And ultimately, even though it was considered goofy science at the beginning, in the end, scientists like data, and we kept putting out data. And then the other lucky thing that happened was that other scientists started to discover what we had discovered, starting in this bioluminescent bacteria, but in terrestrial bacteria, in pathogens, in bacteria that were important to agriculture. And so those data, and those discoveries came along, but we were way out in front. Right? Because that's all we'd been thinking about. And so those things happened together. Princeton gave me resources and fantastic students. And then other scientists, were coming behind us and, fantastic colleagues. And then this field just exploded into being, right? Kind of in a moment, right? And now, what's funny to me is that it's in textbooks now. And of course, that's just the way science, I think, works, is that
so thanks to you, now we have a very brand new way of looking at bacteria.
Yeah, it's just understood now that bacteria are social, that there's all these chemicals, that they do these group behaviors, right? And it's not, it's a very mature field. I still think there's a huge horizon out there, right? We've known about bacteria for 500 and we've known about quorum sensing for 30, so 30 years, right? And so I think that there's lots more room for discovery, but to the beginning of this, when this idea that bacteria could communicate and could coordinate their behavior, it was considered fanciful. And, but there were other moments, even after the quorum sensing field got going, so my gang, first we understood that bacteria talk, to their own siblings. And then we had this idea and we had discovered this molecule where we could show that they were communicating across species boundaries. And that was not accepted. Even after we understood quorum sensing existed, the next sort of moment was, they talk across species. And again, it was like, bacteria can't do that. And like now we know, that they communicate with chemicals, to the human intestines, across domains. And like, I think at each step of this, we and also scientists, we just give bacteria such short shrift, and I don't know why. And I'm guilty of this too. When the gang in the lab brings me a discovery, that we've been working on, like the viruses are listening in, the human host cells are talking back, they're talking across species, they can distinguish self from other, friend from foe. I also am skeptical of that, and I don't know why. I keep thinking like, well, that can't be right, and that I have to be convinced too. You'd think that I, of all people, would give them more benefit of the doubt, but still they keep surprising us. And I think because we just have this 500-year-old dogma that these are ancient, simple critters, that every time we or others discover some miraculous property of bacteria, our immediate knee-jerk reaction is bacteria can't do that. And then we're always wrong. And so that's actually very joyful to always be wrong about that, because I need a job, so they better do some interesting tricks, right? But I do have to say that I'm as guilty as the rest of sort of the science world was still, like, to not realize, or not, I do realize we all came from bacteria. Yeah. Of course, these traits are embedded in them, and why we keep being so surprised, about how amazing they are. I should just be like, yeah, of course they can do that. But I'm always amazed.
Like you said, they act in unison to do some specific tasks, like it is for building a biofilm, something, or sharing food among their own kind, or spewing out a lot of toxins collectively so that the virulence increases, and things like that. And you also mentioned yesterday that about a quarter of a bacterial genome is involved in these kind of collective tasks. What does that tell us? I mean, in terms of...
Right. So the world is huge, right? And bacteria are tiny. Yeah. So whenever a bacterium gives something away to the world, an enzyme, a toxin, whenever it releases something from itself, it's gone, right? And so the only way bacteria can do things, outside of their cells is in these groups. I give you and you give me, and then we all profit. And that's quorum sensing. Okay, so that we get it. It's about public goods, like that I have to, give away an enzyme to metabolize some food that's out in the world, and then I bring up the biteable bits later. These are quorum sensing behaviors, right? Are the ones where you get it. It takes, to be a pathogen, to build a biofilm and cover us with a matrix. It takes lots of cells working together, and then the task becomes successful. So bacteria have a few thousand genes, 2,000, 3,000 genes. And what we now know in each well-studied quorum sensing bacterium that's been studied, somewhere between 200 and 600 of those genes are controlled by these communication circuits, right? So that means when a bacterium makes the switch from going it alone to acting as part of a group, it's got to turn on a quarter of all of its genes, one by one, to the right amount, to the right level. So it's a huge commitment.
Yes.
To stop going solo and to decide I'm going to be part of the group and risk giving all this stuff away, risk turning on all of these genes, but they do it because they succeed. I mean, that's evolution. They would not do it if they didn't succeed, right? But what we get is that quorum sensing's a major part of bacterial life. And then going back to the beginning of our conversation, it's not just about bioluminescence, right? Yeah. Even in the bioluminescent vibrios, yes, one of the genes that turns on was bioluminescence. That's what we could see with our eyes. But now fast forward to the fantastic technology we have now. They turn on bioluminescence and 599 other traits. Yeah. It's just that we couldn't see and measure those at the beginning of this field, right? The bioluminescence was the one thing we could see. But now we get in all quorum sensing bacteria, probably, it's a big deal. It's not a one gene trait. And again, I think one way to think about it, it's the difference between being, a subsistence farmer out in the middle of America where you have to do everything yourself versus when you're in these communities, you can be the car mechanic, I'm the grocer, someone else is the librarian. So they begin to specialize, they take on different traits. And then it's like a city where the bacteria in the community have different jobs, right? They share expertises, if you will, and then the community succeeds because they can profit and specialize from each other's activities, and they don't have to, do it all themselves.
So this fraction of the genome being part of this collective task doing, does it kind of reduce as we go higher up in the evolutionary ladder? I mean, is that...
So nobody has found quorum sensing yet in a eukaryote. Okay, so what we do know is that eukaryotes make molecules that the bacteria detect as autoinducers. So clearly they communicate across domains. So like my intestinal cells make molecules that are very similar to bacterial autoinducers, and then the bacteria respond and they do group behaviors. So it's sort of a one-way conversation. What has not yet been discovered, and maybe it never will be, is actual quorum sensing in a eukaryote, where eukaryotic cells count themselves and change their fates or their behaviors based on a secreted molecule. That just hasn't been discovered yet. And maybe it doesn't exist. I think it might just take time. I hope it takes time for that to be discovered. We came from bacteria, right? So my guess, I'm a little bit brainwashed, but my guess is that there will be quorum sensing mechanisms in eukaryotes. But so far, none of the genes and the proteins that we have identified and others in my field that are involved in quorum sensing in bacteria, those genes do not exist in eukaryotes. We know that much. So if there are molecules that eukaryotic cells use for quorum sensing, they're going to be different.
But bacteria do not have brains or nervous system, right?
Correct. They do
But
not.
They are making calculated decisions, when to act, what to do, and so on. They gather the neighborhood information through chemical chit-chat, sense who is there and who is not by measuring the concentration of the autointroducers. All this information is stored somehow and processed.
It is. They don't store
population density is kind of inferred from, as you said, from the concentration of autoinducers. Transcription factors are initiated. Genes are turned on and so on. all this is working like a computer. I mean, where does this come from? I mean, they don't have brains and nervous system. How does this computing power, I mean, the processing power come about in a bacterial system?
Yeah. So the processing part in a bacterial system is almost identical to the processing parts that happen in your body, right? In your cells. So molecules, accumulate in the environment, and then the bacteria have receptors on their surface. You can think of them as little antennas, that detect these molecules, that are just bumping into these receptors. They bind them, and then information comes into the cell chemically, and genes turn on and off, as you said. That is exactly what happens in the cells of your body, right? Yeah. We call them hormones, right? All of these molecules that tell your kidney cells what to do, tell your heart cells what to do. It's incredibly analogous to what is happening in an individual bacterial cell. But in the end, we do have brains. We do have a nervous system, right? We have all these different organs, right? And bacteria, they do not have that. But they are like the miniature version of what's happening in our bodies, in the cells in our bodies. There's something very different about having a brain, about having consciousness, about having intelligence than what is happening in a bacterium. However, when you look in a human or any organism and in a bacteria, the actual biochemical processes that give rise to life are almost identical. The way you metabolize sugar is exactly the same as the way a bacterium metabolizes sugars. Like we're just not that different at a molecular level.
So, I mean, but at some level, it does appear that there is some kind of, information gathering, storing, and processing going on. Where does that come from? I mean, that is still intriguing to me. That how does that happen? I mean, because at certain point they decide that I must do this, right? How does that kind of decision making happen? I mean,
so in the bacteria, the way it happens is, as we've discussed, the molecules are accumulating, and when the molecules pass a threshold level, so since the bacteria have these receptors on their surfaces, all of the receptors become bound. They're saturated with the molecule. And that is the moment that information comes in. And for the scientists in the audience, it's by a phosphorylation cascade. And in the end, a transcription factor either binds or doesn't bind DNA, genes turn on and off, and then behaviors happen. It's not more complex than that. I mean, it is decision making, but it's simply molecules bumping into receptors on the surface. And when they don't do it enough, the bacteria, if there's not enough molecules around, that the receptors don't have any molecules in their binding sites, the bacteria act as individuals. When the molecules are bound by the receptors, they launch, and it's biochemistry. I don't mean that in a trivial way. And information is chemical, comes into the cell, right, to literally tell a transcription factor to sit on DNA or not and transcribe genes. Genes make proteins, proteins drive behavior. It's actually pretty simple, even though I have spent my life trying to figure it out and I'm not done. We do, at a molecular level, we do understand that it's just chance that a molecule will bump into a receptor, and that has to do with how many molecules are floating around. And again, that is what is happening in your cells, exactly what is happening in your cells as well.
You just mentioned that bacteria also communicate across species. Mm-hmm. In fact, you mentioned that they are multilingual, in the sense that, to be accurate, they are trilingual. That's what you mentioned yesterday. In the sense that they communicate within the species, they communicate across species, and there is also the language which is very genus specific. Mm-hmm. Can you kind of describe or illustrate the kind of quorum sensing that happens in each of these kind of communication channels? I mean,
so I think that the amount of information bacteria need that we understand so far, right? They need to know when a bacterium is in a community and it's going to turn on or off 600 genes. It needs to know who's around, right? So these molecules, what is encoded, it's so amazing to me. What's encoded in these molecules, the amount of the molecules present says something about how many cells are there, but the actual structure of the molecule says something about how closely or distantly I am related to my neighbor. And the bacteria access that information so that they can assess how many cells are around me, and are they my friend or are they my enemy, right? By measuring the amounts of the different molecules that are there. And then they tailor what behaviors they carry out based on the number in the population and also something about the identities of their neighbors. And so we've found, right, that there's molecules that are very species specific. There are molecules that are genus specific, and there are molecules that are unspecific. And so there's a lot of information encoded in these tiny little chemicals.
Now, before we proceed, I just want to understand some basics so that we have a clear picture of what we're talking about. The size of a bacterium is about a few micrometers, right? And I guess this chemical signaling that we talked about through autoinducers happens through diffusion.
Correct.
Right. And the other day you also mentioned that, at least in the case of intraspecies signaling, the autoinducer happens to be hydrophobic, which means the diffusion length is not very much, right? So it gets diluted very fast. So how big would it be a colony of bacteria or the biofilm that they're trying to make? I mean, how far does this quorum sensing happen in terms of dimensions? I mean,
yeah. In the lab, we have standard ways of growing biofilms, that make it easy for us to measure them, right? And so they're tiny, tiny, right? Because that's useful to us. But in the world, the real world, there is a biofilm covering every animate and inanimate object on this earth. You are covered in a bacterial biofilm. The ocean, the top of the water, has waves, but the ocean is actually very steady. There is a biofilm that covers the ocean, right? Like the size can be, it's not limited. And you're right. What you're talking about is quorum sensing. How far can quorum sensing go? Well, it depends on how big the community is. So if the bacteria have resources so they can eat and divide, quorum sensing will occur across a tiny community and a large community, so long as flow doesn't wash away the autoinducers,
yes.
Right? So you're right. It's about diffusion, right? So if, there's a way for bacteria to not be subject to flow, the autoinducers will accumulate as long as the bacteria are growing and eating and doubling. Everything we see with our eyes is covered in a bacterial biofilm. We just don't see it. Your skin, your intestines, these objects, right? There's biofilms everywhere.
So you mean to say that quorum sensing can occur across all kind of scales? I mean, is that...
So long as, right, but remember, so more cells, more autoinducers.
Yes.
Right? Flow, which is ubiquitous in the world, in a river, in the ocean, in a faucet, in your intestine, flow will wash away the autoinducers and then quorum sensing stops. But that's what's supposed to happen. That's how you know I'm alone, right? So long as a environment is, if I would call it static, and the bacteria, right, remember, if they're not eating and growing, they're not going to make autoinducers, right? But so long as they're flourishing and growing in number, autoinducers are tracking, they're building up and tracking, so long as flow doesn't wash them away, as far as we understand, right? But then maybe to the point that you made, the autoinducers that were, right, so these molecules, they encode something about cell number, the different structures encode something about who you are, who I am, my actual identity. But they have also this is the point you brought up, they have really interesting physical chemical properties. So some autoinducers are very unstable. So they have a very short half-life, and they're just not they're delicate, if I can call it that. And so they're around, and then the environment destroys them, and so they're gone. Other autoinducers, you said this, are very hydrophobic. So in fact, they stay really close to the cells, the bacterial cell surface. And so what we think then is those kinds of features give extra information to the bacteria, right? So molecules that come and go are like short signals. Molecules that stay close to the surface are about space. So we think beyond number and identity, other information about the environment, like how harmful is it, or how hydrophobic is it, or how, that there's long range, short range signaling, that there might be way more information in these autoinducers than we've even measured so far, right? Because we just didn't even know how to ask a question about that. But we think we're still missing the boat on these autoinducers. There's more there that the bacteria are monitoring than we know about, simply because we haven't asked the right questions yet.
Though we talk of a threshold being there, threshold number of cell density being there for quorum sensing to set in, we cannot actually quantify. Can we quantify it?
We can. In a lab.
Yes.
In a lab where we have a very controlled environment, we can absolutely measure with great certainty how many cells, how much autoinducer. We can measure those things extremely precisely. Do they do this behavior, this behavior, this behavior? Out in the world, forget it. It's super wild west out there. There's all these other things that are changing that make the autoinducers come and go or destabilize them, or, and so really hard out in the world, which is, of course, why we do these things in lab, is because then we feel we can get the basic fundamental rules. And we hope that we're close to understanding some of what happens out in the world.
So, but is there some kind of a strict correlation between the autoinducer density and the number, the threshold number?
Absolutely. So the quorum that the bacteria count to. So as far as we understand it, each species of bacterium counts to the quorum that it has evolved that's right for it to be successful. So just as an example, if a bacterium has a really potent toxin, counts to the low number. If a bacterium has a really wimpy toxin, it has to count to a higher number before they flip into what I would call quorum sensing mode. And so we think evolution has optimized for each species what is the right number of neighbors that I need for me and my kin to carry out tasks A, B, and C versus tasks D, E, and F. And so it's not like there's one quorum in the bacterial world. Each species over billions of years has evolved with its autoinducers the right threshold number that then these tasks become successful. And so, of course, it depends on the tasks.
You also mentioned that in a given biofilm, for example, like you mentioned the example of about 600 different kinds of bacteria being there in the plaque on your teeth. Each of those species is actually trying to build a biofilm through quorum sensing. And all these 600 species are trying to do the same thing. But finally, there is one single biofilm which encapsulates everything. So whose biofilm is it?
Yeah. Right. So it is so the biofilm on your teeth, that's been studied very well, not by my group, but by other scientists. So as you said, we think there's like 600 species there, and you brush them off in the morning, they're back the next morning, and it is not willy-nilly. Right? There are the initial colonizers, right? It's architected, like a city, right? So there's the initial colonizers, then the secondary colonizers, and they're not all trying to do the same thing. They're all trying to flourish. And I want to be really clear, it is not altruism. What happens is that each species doesn't have all of the enzymes or the proteins that it needs to make the community flourish. So a few species have the ability to attach to the tooth. Other species have the ability to metabolize an important nutrient. Other species have the ability to, say, cover the community with a matrix. And so together, the community flourishes because each of them is carrying out their specific jobs, but none of them has all that they need to flourish, right? And so they, if you will, put up with each other as like neighbors because they have to. And because each of them makes more progeny if they will, in a way, cooperate with each other by, I'm the colonizer that can attach to the teeth. You're the guy that gives me access to this nutrient. You're the guy that keeps antimicrobial peptides away from me, right? And so it is a multi-species biofilm.
Cooperative behavior. That's very interesting to me.
Yeah, but they, if each of those species could do it all by itself, it certainly would. They can't. And so over evolutionary time, they get these sort of architected communities. And that's true in all kinds of different examples, right? Where I donate the genes that I have, you donate the genes that you have, but they're very different, right? And then together, the entire community, well, you get a cavity, but the entire community eats and lives and makes progeny.
Now, bacteria don't care, as you just said, if they're doing good or bad to us, or they're killing the competitors or whatever. They're adapting different strategies and acting different ways for their own survival in the Darwinian sense, right? And they have clearly perfected this art over billions of years. How do you view all your findings from that perspective of evolution, survival, and so on?
Yeah, I think that, it is evolution, right? And so maybe I could step back a little bit. Most of the biodiversity on Earth is in the microbial world, right? So bacteria have had billions of years to evolve different traits. They are so much more different one from another than every eukaryote is to each other, right? And when you look at, if you've ever seen the tree of life, how we make a family tree of your cousins, your aunts, your uncles. We can make these trees of life now based on genomes of organisms. And so we have this huge tree with all these organisms. Everything you've ever seen with your eyes, is just the tiniest little piece of one branch of the tree. Most of all of life on Earth is microbial. So it's invisible to our eyes. And so because they have had so long to diversify and occupy every single niche on Earth, they are optimized, each of them, for their own niche. There's bacteria that live, in these thermal vents in the bottom of the ocean. They live under the ice caps. They live in your gut, but they're all sort of specialized for their environment. And that is evolution, right? And then in some of these cases, they have to work together because literally they get to make more progeny. That is what a bacterium wants to do. It wants to divide and make more progeny. And so for sure, quorum sensing has enabled them because they can carry out these tasks that they couldn't alone. So they get to make more babies, if you will, right? And then this idea of these multi-species communities and biofilms, ultimately, each species that's participating must be getting a benefit in the Darwinian sense, or they would not, evolution would have evolved for them not to interact that way.
Now we have seen, through what you have described, that bacteria, though unicellular, behave much like multicellular organisms. Would you say that on the evolutionary ladder, multicellularity in prokaryotes was some kind of evolutionary precursor to multicellularity in the eukaryotes?
That multicellularity in bacteria, so a biofilm, these communities, it's an analog for multicellularity in eukaryotes. So remember, in bacteria, right, when they make a biofilm, right, so let's just say there's billions of cells in a biofilm. There can be, right? I can take one cell out, right? And it would live and it would divide and it'd be perfectly merrily fine, right? There's something, because each one of those cells has its own genome and has the capacity to make more of itself. Right. That is not true in a eukaryotic multicellular organism, right? So in my body, every cell has exactly the same genome, and I cannot just take my kidney cells out. First of all, not good for me. And also they can't exist, right? And so there is something fundamentally different about a multicellular bacterial biofilm and any multicellular organism. And then the other thing is that when you have a real multicellular organism from an embryo, it starts as one cell and grows into whatever, a worm, a fly, a mouse, me, right? From inception through development, you have one shot, right? You have to go to completion or the whole organism dies. That's not true about a bacterial biofilm. I can stir them up, let them go, and they'll all make a biofilm again, right? And so there really is. So what I think when I think about the parallels between multicellular bacteria and eukaryotic multicellular, I think some of the rules are the same. Who does what? Specializing, taking on different jobs, right? That happens in your body too. But there's really something fundamentally different because each one of those cells can do it again infinite number of times, and the cells in your body cannot.
Yeah.
Right? And then again, also when we talked about having a multicellular, like a multi-species biofilm, bacterial biofilm, every one of those species has a different genome, set of genes. In my body, no matter what fate a cell has taken on, they all have exactly the same genome. And so all the cells in my body have a vested interest in all the other cells prospering. That's not true in a bacterial biofilm, right? They're out for themselves.
But what I meant is during evolution, was there kind of a switch that happened from one kind of multicellularity to the other, or it happened in some kind of various steps or various kind?
Well, so for sure we came from bacteria.
Yeah.
Right? That's evolution. And so somehow some of these principles that are embedded, let's call it in a biofilm or bacterial multicellular community, some of those principles rose up through evolution. But what the moment was where we got real multicellular organisms,
we don't know.
I don't know. Okay. Yeah.
Right. Now, you have not talked about the new electrical mode of multicellular, intercellular communication that has been discovered by this University of California, San Diego scientist,
Searle. Yeah.
Using much like brain and nervous system, using potassium channels and action potentials and so on. And this would not have the limitation of diffusion length, right? And they would be much more powerful and so on. Probably quorum sensing would be over a longer range than otherwise. So, I mean, do these kind of electrical signals lead to different kind of quorum sensing and different kinds of collective behavior, you think? I mean,
yeah, so I don't think the girl thinks of his electrical signaling as quorum sensing, right? Because you're right, there is no diffusion, right? I think he's much close. His work is much more about how did electrical signaling, which happens all over my body, right, in neurons, how he's trying to figure out the principles of that and how that might relate to higher organisms, right? But I don't think he's thinking about that as quorum sensing. I don't think that's what that is. It's much more directed signaling, and maybe the principles for how neurons signal to each other. Like he'll find the skeleton principles of that in his system, the same way I hope I'm finding the skeleton principles of multicellularity and communication in my system. So his work is wonderful.
So as I said, that you also recognize being an evolutionary precursor to brain and nervous system and so on.
And there's and I think that. Yeah, and we also know, bacteria do mechanical signaling, like touch signaling, and of course that's what happens in your body. Your liver stops growing at a certain point because all the cells touch each other. And that's mechanical. Bacteria have those kinds of mechanosensors, and they do mechanical things. So again, I think these, maybe it shouldn't be surprising to us that these mini versions of us, of all these different important processes that give rise, to a fascinating multicellular organism, that the skeletons of those simpler skeletons exist in bacteria. And girls, at UCSD, his is about electrical impulses or signaling.
But what other kind of physical communication channels exist among bacteria?
So bacteria sense all kinds of...
You mentioned touch.
Yeah, so there's touch, right? He has electricity, I have chemicals, but bacteria... Their membranes are decorated with receptors for all kinds of environmental stimuli. It's, I'd like the world to think it's just quorum sensing, but it's not. They sense how much nitrogen is there. They sense how much food is there, right? And they do this behavior called chemotaxis, where if there's nutrients, a gradient of nutrients out there, they can change the direction of their swimming, just like, so that they go toward a nutrient and they can run away from poison. It's sort of like your grandmother's baking cookies in the oven, and you make a beeline for it. Like bacteria invented that. It's sort of like they have a sense of smell, right? They have a sense of touch where they feel these mechanical processes. And so again, they're just masters of sensing and responding to many, many inputs in the environment, integrating that information, and instant to instant to instant, the bacteria behave appropriately based on so much more than quorum sensing. So quorum sensing is just a part of that incredible repertoire of sensory, information intake. That they are the best at that. Yeah.
Now, strategies for Darwinian survival would include tricks to interfere with quorum sensing. Okay, interfere and probably one species trying to silence the quorum sensing of the other, and so on. How have you used this principle of quorum sensing quenching, as we call it, to develop, possible therapies of the future, approaches to potential therapeutics to ward off diseases due to bacteria, which are now faced with antibiotic resistance and so on and so forth.
Right. So early in this field, so once the field exploded out of bioluminescent bacteria into pathogens, and of course my gang was part of that, what we recognized very early on is that if bacteria can't talk or they can't hear, they cannot turn on their repertoire of pathogenesis genes. They can't make a biofilm. And so we, the scientists, were like, holy moly, right? If we could make synthetic strategies that can shut down quorum sensing on demand, those could be potentially new kinds of therapeutics or applications for industry, for bioremediation, for agriculture. And so many people in the field, myself, my gang included, we all started to take what we had learned from our basic learning about quorum sensing, about the molecules, and we made lots of synthetic strategies, right? Like anti-quorum sensing molecules. We would try to make it so the bacteria couldn't even make the autoinducers, to try to shut down quorum sensing in harmful bacteria in the hopes of making new kinds of applications. And it worked pretty well in the lab. We've made many different strategies that shut it down. But then there was a point, to the point that you just made, where we thought, at that point, we've known about quorum sensing for a decade. Bacteria have known about quorum sensing for 4 billion years. Maybe they already invented this. What the heck are we doing in the lab trying to make these strategies when the really good ones probably exist in nature? And also, since bacteria have been duking it out and fighting against eukaryotes, for all these time, the anti-quorum sensing strategies that exist in nature have been tried and tested over evolutionary time, not Bonnie Bassler lab time. And so the ones that exist out there, they have to be the ones that are the best. And so we and others started going out, it's sort of like panning for gold, right? We call it bioprospecting, right? That we and others have gone out and tried to learn, again, in the same way, just by basic making discoveries, learn about the strategies that exist out in nature and bring those back into the labs and use those as the building blocks, inspired by these bacteria. And then maybe we could tinker with those and make them even better, because they, those strategies that exist in nature are good for in nature. We need them to be safe. We need them to be effective. We need to, to sort of tailor them for purposes for humanity. But we think that's the gold mine, is to go out there and get them. And it's really fun in a way, because these bacteria, They eavesdrop on each other. They trick each other. Like one guy makes, molecules that it doesn't bother responding to. But of course, if the neighbor detects it as autoinducer, it's going to make all these goodies, and then the competitor gobbles it up. Some bacteria make autoinducers, and the guy next to him in the dirt, these tend to be soil bacteria, they cut their autoinducer. They have an enzyme, and they cut their autoinducer in half.
Okay.
So the one guy is trying to talk and tell its kin, we should be doing these things, and the guy next to it in the dirt makes it mute, so it loses competitive advantage. They make cheater molecules. They make inhibitors. There's also bacteria that make an autoinducer, that the autoinducer for me is a quorum sensing autoinducer, but if you detect it, it's a poison. So if you try to listen in, you get killed. So they make misinformation, they confuse each other. And so we're all lots of people in my field now are trying to get those strategies and actually tame them, if you will, so that they're useful in applications.
That's fascinating.
Yeah, it's really fun. We should have thought of that so long ago instead of trying, the first ones we all just tried to make, like with synthetic chemistry.
Right.
And we should have long ago thought, oh, come on, the bacteria beat us on this too.
Yeah, sure. Yeah. So, that's really, have you been successful in actually?
Yeah. So, it takes so for sure and I'm not there, there's lots of members of my field, hundreds of labs work on quorum sensing now. Right? We're just a big flourishing field. And so many different scientists are having lots of good luck, not good luck, they're making their luck. And that these strategies work really well in the lab, right? But the what am I call it? The path from an academic lab, me and like 9 22-year-olds, right, to making at scale a medicine that's going to go into people and is safe and effective and potent, that's a slow, super expensive path. And then to the point that you already brought up, it's a new way of thinking about how to treat bacteria, right? Like it's behavior modification instead of killing or slowing bacteria, killing or slowing bacterial growth, which is what a traditional antibiotic does. And so I have to say, sort of disappointingly, it's slower getting there than we thought it would be. And I appreciate why, right? It's a paradigm changing idea for medicines. So I think the applications that are going to happen faster are the non-medical ones. So for example, dangling anti-quorum sensing molecules off of like say plastics to make, for example, you could make infection resistant tubes that would go in the body, paints so that bacteria can't gunk up cooling towers, disinfectants so bacteria can't, get on surfaces in hospitals. So the sort of applications that don't go into the human body, they're happening faster, like in agriculture, for example, in fertilizers and especially in bioremediation, things like that. And so I think those are happening faster. And the hope in the field is that as those applications flourish, there'll be more and more impetus to actually move those to the medical front. Yeah.
But the kind of antibiotic resistance that we're seeing in the current antibiotic in the market and so on, that kind of resistance, do you think even anti-quorum sensing molecules would be faced with at some point of time?
Yeah, sure. So, of course, antibiotic resistance, one of the most globally important, urgent challenges on Earth, right? And there's not very much work being done to confront that. And so let's say we could make a really potent anti-quorum sensing molecule that was a medicine. The bacteria will get resistant. We have learned that lesson. But the idea in the field is that because an anti-quorum sensing molecule doesn't kill bacteria, right? So that the only bacteria that can live and grow are the ones that are resistant. Since we're just doing behavior modification, we're making they can't talk, they can't hear, giving time for the immune system to wipe them out. The idea is that behavior modification molecules, like an anti-quorum sensing molecule, would simply have a longer shelf life, giving some young scientists more time to think up the next Achilles heel that bacteria have and develop the next strategy. But we're not foolish enough to think that they will not get resistant. We just think it's going to be a softer selection for resistant. And so maybe they would last longer as therapeutics than what we have right now. That's a fantasy.
Let's talk about your new work, the fascinating work that you talked about the other day. Viruses, the acellular organisms more primitive than bacteria, basically hijack functions of higher organisms and infect them and so on and so forth, for their own survival and growth, obviously. But in recent years, you and your colleagues have made this amazing discovery that viruses that attack bacteria, called phages, actually eavesdrop on bacterial quorum-sensing molecules and try to interfere with their process and trigger them to kill themselves, so to speak. How do viruses actually do this? I mean,
So viruses are the ultimate parasite. Right. So there's a debate about whether viruses are alive or not. I don't think that's that important. Right. So viruses, their strategy is they have to be inside a cell, and then they hijack cellular components to make more of themselves. Right. They're so insidious. Right. And so they're these primitive things. They're not even organisms. And so viruses, that's what happens. They infect human cells. They hijack the components of human cells. We all been infected by viruses. But bacteria are bombarded by viruses also. And for historic reasons, we call viruses that infect bacterial cells phages. We call viruses that infect higher cells viruses. But they're sort of the same kind of entity. And so one of our most recent discoveries, and this was a graduate student, Justin Silpy, in the lab, what he discovered is that some phages that infect bacteria, they have genes that encode quorum-sensing receptors that bind autoinducers. And so so they're eavesdropping, right? So what happens is as the host bacterial cells grow, they're infected by the phage. And if a phage is going to decide to replicate, kill its current host, and get out into the environment, if there aren't any other bacterial cells there, that phage is a goner because they have to be inside a host cell. And so what these crazy phages have done is by evolving, or capturing receptors for quorum-sensing autoinducers, they can detect when the hosts are at high cell number. So if they replicate and kill their current hosts, there's many more hosts around, right? And so it maximizes transmission to the next cell. And so they're eavesdroppers on host quorum sensing, and they only kill these host bacterial cells when the host bacteria are at high cell number. And it lets them propagate in a way they could never, it's basically they're hedging their bets. They're not like replicating and then they get out and there's nobody there to infect. They ensure by listening in, if you will, to the quorum sensing molecules, there's a lot more victims out there. Now's the perfect time. It's the optimal time to kill. Exactly, because I'm going to spread. And that's, of course, what a virus, that is the viral strategy, spread to new cells. And so that means, it's just awesome, amazing. Like, why didn't we think of that before, right? Like, if you think about it from the viruses or the phages perspective, I should say, once Justin got this discovery that they kill the cells, at high cell density, it maximizes transmission. Again, never giving these microbes their due. We're like, of course it's like that. That makes perfect sense once you know it, right? And so we think that that's really a fantastic moment in evolution. We've now got many, many cases of phages that have quorum-sensing genes on them. And so the one part of that is that it's changing the way we've thought about phages. But the other part of that is it means that quorum sensing is spanning domains beyond, bacteria and beyond bacteria to eukaryotes. Phages are way more different than bacteria are from eukaryotes. And yet quorum sensing crosses that boundary now, right? These little molecules, right?
Tell me, you mentioned this, quorum sensing probably happens across all domains, but do viruses or phages themselves have their own kind of quorum sensing?
They do. So there's the first example of that is not mine. It's a scientist named Rodom Sorik. Mine is a phage bacterial quorum sensing interaction, the one that I described in my talk and that we're talking about here. A colleague of mine, Rodom Sorik, he's in Israel. What he found was that there was phage quorum sensing. And so what he showed in this beautiful paper was that, so when a phage decides to leave its current host, it wants to infect another cell, but it cannot infect a cell that already has a phage in it.
Right.
Right? That's that, because it's already no room at the inn, right? And so what Rodham discovered with his group is that the phages make quorum-sensing molecules, and then if there's a lot of quorum-sensing molecule around made by the phage, it means everybody's infected, and so the phages will stay dormant. But if there's not very much quorum-sensing molecule around, it means most of the cells, the host cells, are not infected, and that's when the phage strikes.
Okay.
So he found phage quorum-sensing. The first example of that, we found host phage quorum sensing. And then what we also found recently that I didn't talk about in my talk is we found phages that make autoinducers that the host listens into. So what I mean by that, now there's phage-phage communication, there's host-to-phage communication. That's what I talked about yesterday. And now recently, and we haven't published this yet, there's phage-to-host quorum sensing, right? And there's going to be many more derivatives of that, right? Because this is just the beginning of what I think is a brand new field.
Yeah, as you just said, I mean, each host has multiple kind of,
They're littered with phages.
phages, right? Now, there's also competition, as you said, of which phage will actually get into the host, right? I mean, what kind of strategy do they adopt to?
Right. So it was always thought, so if a host has two different kinds of phages, remember you can only have, you can't, right? So it has two different kinds of phages. When those phages decide they're going to replicate and kill the host and move on, it was always thought it's just a sprint. Whichever phage is better at getting more host components, whatever, that phage makes more of itself at the expense of its competitor. That's true in many cases. But now that we realize that these phages have these little modules that allow them to listen into quorum sensing, what they can do is if one phage can listen into host quorum sensing and the other co-resident phage can't, by having an extra sensory input, now what we realize is that these little quorum sensing circuits on the phages allow them to better compete with one another. And so the one phage is kind of, sleeping, because it's not listening into the host cell density, and the second co-resident phage just wins in the competition. And so we think that these modules, I'll call them modules, these eavesdropping modules, they help the phage decide when to kill the host, but they also give a leg up on a competing phage, right? That they're very worried about, right? Taking host resources. So we think these little modules are super versatile.
So
right?
Their autoinducers are somewhat different from those in bacteria, are they?
No, they make autoinducers that so far that we've discovered, they are similar to the bacterial autoinducers, and they trick the host into doing things the phage wants them to do because the autoinducers are so similar.
I see. Okay.
Yeah.
Now, of course, in this billion-year-old phage bacterial war that's been going on, bacteria have, of course, evolved their own self-defense mechanisms, including the what you mentioned yesterday, the CRISPR-Cas mechanism, or restriction enzymes, or abortive suicide, so that they protect the neighboring cells, a kind of collective behavior, social behavior, if you may say. Yeah. Now, recently I read about discovery by a Lithuanian group of a new kind of kamikaze mechanism, called SPARZA. And it's something like Justin's acronym. Actually, it's quite convoluted in that. It's called Short Prokaryotic Organoid DNase Associated. I was attracted to this news item because SPARZA in Hindi, in the Indian language Hindi, actually means competition.
Oh.
Okay. Now, this has been recently published in Cell Research. But there is actually an ongoing arms race, right? Between bacteria and their antagonist phages, with phages evolving their own strategies. Would like to hear something more about this.
Sure.
What's happening in this?
This is what we call bacterial phage defense and anti-phage defense. This is, you already said it, it's an arms race. So bacteria as we talked about, are bombarded by phages.
Yes.
Right? They are the enemy, right? And so, phages try to capture host components and hijack all the components. And so bacteria have evolved strategies to restrict phages from doing what phages want to do, which is hijack the components, kill you, and move on. And so the first one ever recognized was what are called restriction enzymes, which is an anti-phage defense mechanism that bacteria have, but which scientists discovered. They got the Nobel Prize for it. This was in the 70s or 80s. And then restriction enzymes were applied to use for cloning, revolutionized molecular biology. The next one was the famous CRISPR. Which is now in the news,
yes.
which is an anti-phage. Originally, CRISPR is a bunch of bacterial genes that when a phage invades the bacteria, it tries to kill if you will, it cuts up the phage genome, gets rid of them. Right? Now CRISPR, of course, is being used for genome editing. It's all the rage. It's another Nobel Prize, right? It's revolutionized our ability to manipulate genomes. And so then it was thought it's all solved. There's these two, there's restriction enzymes, and now we have CRISPR. But then those inspiring discoveries got people like this Rotem Sorek that we were talking about. He's very much in the forefront of this field to figure out that scientists have now been looking for other bacterial defense mechanisms against phages. And at last count, I think there were 150 new mechanisms that have been discovered. It's a field, right, of these scientists discovering all of these mechanisms that we never knew were there, that are about, like the one you just said, like that are about bacteria defending themselves from their ultimate enemy, which is phages. So, and then what they often do at the end, what will happen is that the bacterium will launch this defense mechanism that will restrict or kill kill is a funny word, that will, somehow restrict the genome of the phage. And then that bacterium kills itself. It commits suicide so that the rest of the population can live and the phage can't spread. Right. And so it's amazing. Right. And remember, the same way restriction enzymes and CRISPR, which are phage defense mechanisms, revolutionized applications, you can imagine, right, going forward, that these other 150 now other mechanisms could be eventually, worked into new kinds of applications that are great for humanity, science and humanity. Yeah, it's a huge new field that nobody anticipated.
But conversely as well, since phage have their own kind of defense mechanism, you can even conceive of phage therapies to get rid of pathogenic bacteria.
Absolutely. Right? So phage therapies had a moment a long time ago where scientists thought, phages, what do phages do? They kill bacteria. And so couldn't we use phages as antibiotics, right? And so people worked on that long ago, but then the bacteria got resistant. And so they fell out of vogue, which is right. And then they kind of have a bad reputation, phage therapies. But now this whole world of phages is with CRISPR, with all these anti-phage defense mechanisms, right? It's all coming back to the forefront. And there's huge amount of renewed energy in making much more sophisticated phage therapies than were made 40 or 50 years ago, using what we know now. And again, to the point that you made earlier, we have this global problem with antibiotic resistance. We need to bring any kind of arsenal forward that we could. And so I think that phage therapies are now coming back into the forefront in terms of the potential for them to be really useful antimicrobials. Yeah, it's a brand new area of study. And again, that partly it's brought about by like us having genomes, right? That beyond the bacteria being super diverse, we talked about that across the earth, the diversity of phages and phage genes and the sort of metabolic ingenuity that exists in these phages, right? People are just now starting to harness some of that for humanity. It's an amazing time.
But in a human system, there's this actually three-way war going on, actually, right? Mm-hmm. There is this microbiome trying to fight the pathogenic bacteria. Then there is this phage trying to kill the pathogen, as well as the microbiome itself. So how does this play out in a human system, and who wins? And how do you make one win against the other?
Yeah. Okay, so the question that you just asked, how do you make one win against the other? That's the Holy Grail, right? And so, right, it's remarkable. You have this magical microbiome that most of it just takes up the real estate that keeps pathogens out. But when the pathogens get in, you have your human immune system, you have your microbiome, all fighting against these pathogens. Now we know there's these phages that are in there, part of this fray, if I will. And, who wins? Sometimes the phages win, sometimes the human wins, right? Or sometimes the microbiome wins, and sometimes the pathogen wins. And of course, the goal for us is to learn enough about that by doing these basic curiosity-driven, making discoveries that and we can't do it yet, that we could make who we want win, which is probably the humans and the microbiome more. Right? But I don't know how it really works in a human. Right?
So, what I meant is, which domain do you look at first or most strongly? The microbiome or the phage or the...
So what's lucky is that there's lots of scientists interested in this. So for me, if you ask me, what am I going to look at? I'm going to look at the bacteria and the phages. That's what I'm trained for. That's where all my discoveries are. That's what I'm set up to do in the lab. And I depend on colleagues that work from the microbiome side to be informing me and my team, so that we can make connections. And then they too depend on immunologists who study the human side of how the microbiome gets to be there and how the human tolerates it, right? To be giving them information. And so what's so fantastic about science, right, is that we publish our work and we make everything freely available to one another. So I don't have to do it all, right? But what's great also about science is that there's so many scientists interested in this complex consortium, right? And that we can depend on each other to give us information that we can't get ourselves, because, I have 10 people in my lab and we are not experts at the microbiome. We're not experts at immunology. We're experts at quorum sensing, right? And so, but I think it's quorum sensing. The group gets more collectively, these scientists, it's one of the greatest things about science. But thank you you're a wonderful interviewer, and it's very lucky for me. I'm so thankful to the TNQ Foundation for inviting me here, because it's so lucky for me to get to share my science in all these different venues across India. So thank you for having me. Yeah, thank you.
Thanks.